So in continuation with the previous presentation, so in this presentation, I'm going to explain you about the remaining part of the basis of immunology plus the antigens. Okay, so in this the combination of so last class what we discussed is active and passive immunity. So in this session, and there is one more part, it is called as the combined immunization. So what is there in the combined immunization? If the patient is very serious and they need some antibodies, so those cases what we can do, you can administer both antigen as well as antibody together. So the antibody, it will recover the disease, and the antigen what we are administered either in the form of vaccine or toxide are either attenuated or killed. So what will happen? That will be producing a sufficient memory cells to produce or to protect the individual from to produce the memory cells. So this is what happening in the combined immunization. Okay. And in this case of a combined immunization, both immediate protection as well as the lifelong, long-lasting memory that will happen with this immune system. And one best example is tetanus immune globulin. This is one arm, and tetanus toxin in the other arm. Either it may be in the combined form or it may be in the two different doses you can take it. So one, the toxide, it will be producing memory cells and it will be protect the individual. And the already available immunoglobulin that will be producing as an antibody, and it will be protect the individual from the disease at immediate action. And local immunity, it is the one that refers the protection given to the potential site of entry of the pathogens, and mainly prevents the entry of pathogen with the skin. And immunoglobulins, namely immunoglobulin A, which is involved, which is produced by the plasma cells and present on the mucosal surface. And herd immunity is the one, the overall level of immunity in a community is around 80% of an individual population. There are some individuals, some group of populations, no, they don't get affected by a particular disease. And this is the reason for relevant control of the epidemic. Epidemic is the one in a particular region, people, they may get affected or they may not get affected. So that is called as herd immunity. So this is by nature. When herd immunity is become low, an epidemic is likely to occur. And now in this, the remaining part of this presentation, I'm going to explain you about the antigens and its various, the properties of antigen. So antigen, it can be defined as any substance which is when introduced into the body, it evokes immune response and reacts with the products of immune system like antibody in a specific and observable manner. So this satisfies the two distinct immunological process, or it may be a property. One, it is called as immunogenicity. So the way it reacts with our own immune system, and antigenicity, this is the reaction between the immunological products, which is due to immunogenicity factor. And immunogenicity and antigenicity. Immunogenicity is the ability of the substance to induce the immune response, and then later it may be treated either humoral or a cell-mediated immunity. And here the B cells, it combines with the antigen, and then later it is produces plasma cells and memory cells. That is where we discussed in the previous presentation. And plasma cell, it is the one that is able to produce an antibody. And the T cell mediated or cell mediated immunity, here the T cell mediated along with the antigen presenting cells, and where the T helper cell CD4 or CD8, this together it joins together and it treats the antigen, and later on it also produces the memory cells. Antigenicity is the ability of an antigen to combine specifically with the antibodies or bind to the T cell receptor on T cells and evoke a specific immune response. So a substance which induces a detectable immune response are appropriately called as immunogen. An immunogen can trigger an immune response. Antigen in that response on all molecules having immunogenicity is called as all molecules having immunogenicity, it also showing an antigenicity. So all immunogens are antigens, very well you can say. Despite the fact that all antigens are recognized by specific lymphocytes, or it may be the produced antibody, but not every antigen can evoke an immune response. So when antigen, it does not evoke an immune response. So by the time the phagocytes, monocyte is just getting phagocytosed, and then later by presenting some chemical substances on the antigen, and then it causes an immune response. And antigens, those are capable of inducing an immune response, are said to be immunogenic. And this is called as an immunogen. And all the antigens are not immunogen. An antigen, it may enter when it is not producing or it is not reacting or it's not functioning our immune system to produce some antibody, and that is not an immunogen. And hapten, this is one more important part. They are low molecular weight substances and it is incapable of inducing antibody formation by themselves. But when it combines with a specific carrier protein, and it will be able to produce antibody. So they are called as, it is non-immunogenic, but it is an antigenic. So antigenic is the one it is able to combine with and a produced antibody. So they can become immunogenic when it may combine with a larger protein molecule or as a carrier, like even bentonite or a latex. So this is what is a complete explanation about the hapten and carrier protein. So hapten is a molecule, it itself, when it enters into our own body, the immune system, it won't recognize this haptens and it won't produces any antibody. The same hapten, when you combine with the carrier protein, and you make it the molecule like this, and when that you are introduced into our immune system, it acts as a immunogenic. So what is this immunogenic? It reacts with our immune system either through B cells and T cells, and it will be producing enough amount of an antibody. And that antibody, it will be producing either plasma cells or memory cells, and later that antibody, it combines with this the antigen molecule, and so that it neutralizes. So this is what a hapten, incomplete antigen. It itself, it won't act as an immunogen, but when it combines with some carrier protein, it acts as a molecule, or it will be acting as an immunogen. And the types of hapten, there are two types of hapten, simple hapten and a complex hapten. What is the simple hapten? It is univalent. The hapten on antibody complex, it cannot be visualized, as it is believed that the polyvalency, maybe in the next slide, I'll explain you in detail what it is, the polyvalence. Assume an antigen, it is containing only one univalent the antibody binding site. You cannot see a precipitation reaction. There is a reaction, the antigen and antibody reaction. If antigen and antibody, it combines together, the reaction, there are various types. One particular reaction is called as the precipitation type. So if you want to see that the precipitation reaction, if the antigen, it is containing more than one the epitope or the haptens, so then you can see a precipitation reaction. So to observe the precipitation reaction, the molecule should be as a polyvalent. So what is that polyvalent? Antigen will be combining, or it is containing more than one epitope. All the epitopes, when it is combined with the antibody, then you can see a precipitation reaction. So epitope is one more important part in antigen. Any antigen that will be containing epitope. Epitope is the region where the antibody it goes and binds. And it is the smallest unit of antigenicity, and consisting of 4 to 5 amino acids, or it may be containing monosaccharide residues. It is having a specific chemical structure and electric charge and steric configuration. So because of the specific chemical structure only, the produced antibody, it will be finding a specific target. Antibody which is produced against one particular epitope, it never reacts with the other epitope. And it is capable of sensitizing either T cells or B cells and reacting with the specific site of even T cell receptors or an antibody. So that is what I was explaining in the previous presentation. And combining area and antibody towards an antigen to an epitope, that is known as the paratope. So epitope and paratope, it determines the specificity of the immune reaction. The antigen is antigenic mosaic. An antigen such as bacteria or virus, it carry many different types of epitopes on its surface. And for each epitope, it will be producing different antibody. And presence of similar epitopes on the different antigens that are responsible for antigenic cross reaction. Possibilities are very less, but when you contain some antigens, no, it is containing similar epitopes. So the antibody produced against one epitope, it may go and combine. So that is called as the antigenic cross reaction. So this is what a typical antigen, the yellow color one is a one particular epitope, and the other one, the antibody 2, it goes and combines in this particular position. This is one more epitope, and this is a third epitope, and this is fourth epitope. So if antigen, it is combining more than one epitope, the produced antibody that will be reacted that combines with this antigen and it causes precipitation. So to observe precipitation reaction, that is called antigen antibody reaction, the antigen should contain more than one epitope. So if more than one epitope, for each epitope, there will be one antibody, it will be produced. So that is what we discussed once, a polyclonal antibody and monoclonal antibody. Monoclonal is the one, only the antibody will be produced only against one particular epitope, and polyclonal antibody is the one, the antibody will be produced for a different epitopes of the same antigen. So this is what the epitope region. Okay, so that is why I said whenever you draw antibody and that will be binding, this is the region that when you make it as an inverted Y shape, and the region where it binds the epitope, it's a specific region. The epitope, it won't bind in some other part of the antibody. Maybe in my next presentation, I'll be explaining you antibody in detail. Even by the time you can understand how why this is the specific region antigen or the epitope, it goes and binds and it gets neutralized. And polyclonal antibody, it is containing multiple clones of antibodies which is produced to a different epitopes on the same antigen. For example, if there are 4 epitopes of antigen is present in one particular antigen it enters, there will be 4 different antibody will be produced. All 4 antibody will be reacting with the different epitopes, so that the neutralization, it will be quite faster. And that's what it is called as the polyclonal antibody for a particular antigen. For different epitope, all epitope will be producing different antibodies, and the produced antibody, it combines with its own target and then it will be neutralized. And the epitope, it may be of other type also it is there. One, it is called a sequential epitope, or it may be a linear epitope. Epitopes may be present in a present as a single linear sequence of primary configuration. They are recognized by the T cells. And some epitopes, it may be non-sequential. This epitopes, it will be found in the flexible region of different sites of peptide chain. And during folding, it will go in different places. So we discussed in detail about the proteins during your MBC, medicinal biochemistry. So during folding, this epitopes, it will go on there in the different location. And if the epitope, it is in the non-sequential, mostly that is treated by the B cells. Maybe this you can understand better in this image. So assume this is during folding. Sometimes the epitope, it will be present in one particular location where it will be as a linear. And during folding, in the same, it may happen in the discontinuous epitope, where the red color one, if you see, it's a non-linear or non-sequential epitope. One epitope will be there in one particular portion of your protein or a peptide chain, and another epitope, it will be present in the other epitopes. See, the antibody, it takes the responsibility of combining both epitopes, and so that it makes the antigen, it is getting neutralized. More than this image, still you can understand better in the next slide, where if the linear epitope, how this antibody, it makes the bond, it will be as a continuous region. And if the epitope is a discontinuous or non-sequential, see that the paratope, this the epitope binding region, it makes the binding like this, and so that it causes a neutralization. So this is what that antigen antibody binding, and that's the reason why I told whenever you draw antibody, the antigen, it should bind in this particular region, not in the different location. And then types of antigen, this is one more subdivision or classification you can write based on the ability to carry the immunogenicity or antigenicity. This antigen is divided into 2 types. Either it may be complete antigen or incomplete antigen. So what is that incomplete antigen? Is nothing but it is the hapten. So that is what I was explaining you in the previous slides. And the types of antigen are also divided into the 3 more types based on the origin of the obtaining. The first one is microbial antigen. And these are structural antigens of microbes. It may be somatic O, many cells other than reproductive cells, other than germ cell lines, that is called as the somatic antigens. And even it is present in flagella, maybe one which is caused typhoid. Salmonella typhi flagellar protein is responsible for causing the typhoid. So flagellar, capsular. So that antigen, it is called as a K antigen. It's like every part of microbes, it is containing different antigen, and those antigen will be responsible for producing a specific antibody, and that antibody you can elaborate in detail. And apart from this, and even you can elaborate in detail about the the blood group antigens. And already I think you would have studied in your HAP. So that is nothing but blood group. Now we have a ABO system. In our chromosome, we have a gene of A, B, and O. So which allele it is dominant during our when the chromosomes are combined from father and mother. Assume it's a A and A. If it is combined, the blood group of an individual or the child, it will be A. Or we have three alleles like A, B, O, and which allele will be dominant at the time of conception, and that will be the blood group of an individual. So that is why it is not necessary the parent will be of one group and the child it may not be the same group. It will be of any type. So parent may be A and mother it may be B and the child it may be O group. That is why the allele, what kind of allele it is producing. So whatever that blood group of blood group it is also containing some antibody. So in RBC cell, there is a specific component called antigen. And when you take a blood group of one individual which is containing specific antigen, when you introduce into another individual, they containing, for example, I'm a A group and I'll be containing the antibody B. When I receive the blood group B, what will happen? The antibody which is available for a B group, it will react and it causes the RBC lysis. So that is why while administering the blood group, the matching is very important. So if you need to elaborate in detail about blood group ABO system, and little bit you can elaborate. In RBC, what is the present substance is called antigen, and that antigen is also responsible for producing antibody, and that is why a specific blood group only it should be transferred to another individual based on the blood group matching. And as like blood group, even our own the tissues. Okay, one more we are going to discuss in detail about histocompatibility complex, and every tissue also it is containing some amount of antigen. So our body, because of tolerance, the antigen what it is present in the tissue, it won't produce any antibody. Whereas my tissue or my organ, when I take and transplant to someone else, okay, the my antigens what it is present in the tissue, and the received individual, okay, the recipient, they starts producing some amount of antibody against the organ they received. So that is called as a transplantation antigen. So that is why you have to go for immunocompromised or the immunosuppressants you have to give those individuals those who are received organs from another patient. So that is why the compatibility matching. So they are finding what kind of antigen the patient they can accept. So that is why closely blood related people, when you take that antigen will be of more of similar, and the organ or our own immune system will be treated as even the received organs will be as a self antigen. So it won't produce any antibody. So based on the content you required or mark you required, you can elaborate about this blood grouping system or transplantation system, or else what we discussed in brief manner you can explain. And then exogenous antigen is the one, the antigens present outside the cell. It includes the allergens and parts of microbes, not necessarily the whole microbes. It may be a part of microbes, even drugs, and especially protein-based drugs and pollutants. These are all also act as an antigen. And then other types of antigen, there is one more type, it is called an endogenous antigen. These are generated within the normal cell as a result of normal cell metabolism and because of viral or bacterial infection. And autologous antigen, they are belong to host itself, hence they are ordinary and it is a non-antigenic. So this autologous antigen only, it is responsible when you go for the organ transplant, and this antigen will be identified by the another individual and they starts producing antibody. In autoimmunity, the over functioning of our own immune system. So what will happen? And sometimes suddenly it starts producing the antibody against our own antigen, and that is called as the autoimmunity. And antibody or immune reaction against our own self antigen. So normally not found in the circulation or in the tissue fluid. And the types of antigen based on the immune response. So one, it is T cell dependent antigen. Here the B cell, it cannot respond to these type of antigen and without the co-stimulatory mechanism with the T helper cells. Whereas a T cell independent antigen, these antigens it's intrinsically activate the B cells and to proliferate and differentiate without a T cell stimulation. This is based on the type of the reaction. And the other type of antigen, one more, it is called as a super antigen, antigen that activate the very large number of T cells directly without going for antigen presenting cells. That is what I was explaining in the previous presentation. And one example is Staphylococcus aureus. And the determinants of antigenicity that completely depends on the foreignness, how much it is foreign, and that much it will be producing the immune response of an antibody. Where antigens are foreign to the body, induce an immune response, and normally immune response it won't develop against self antigen due to the self tolerance. And sometimes autoimmunity develops due to the breakdown of the homeostatic mechanism. And one best example is reported for this autoimmunity. The lens protein of an eye, it maintains the intraocular pressure in the eye and refractive index. And this is the one common antibody that is produced against our own the lens protein or an antigen. And and the determinants of antigenicity, it is based on the size. Higher the molecular weight, higher will be the antigenicity. If molecular weight is greater than 10,000 Dalton, it may be mainly antigenic. And hemocyanin's molecular weight of 6.75 million is highly antigenic. And adsorption with bentonite or kaolin, it makes the even low molecular weight substances into an antigenic substance. When the molecular weight of a compound, if it is less, it won't behave as an antigenic. So, but when you combine with some material and it starts behaving as an antigenic material. And some substances, it bind to the tissue proteins and become antigenic. So one example is penicillin, formaldehyde, and picryl chloride. So that is what the penicillin allergic reactions. So once we discussed in incompatibility, some individuals are allergic to penicillin. So this penicillin, when combines with some tissue substances, and it's the tissue it is identified it is an antigen. So against penicillin, immediately the antibody it starts produces, and we found with some allergic reaction, and it may be mild to severe in to anaphylactic reaction. And the determinants of antigenicity, the other factor, the chemical nature, and higher the structural diversity, and more will be the antigenicity. And for example, protein is the highest producing an antibody or immune response, followed by polysaccharide, followed by lipids, and followed by nucleic acids. So proteins in combination, about 20 amino acids or more amino acids, it will be more antigenic than polysaccharides of 4 to 5 monosaccharides. And gelatin, it is being it's unstable and it is non-immunogenic. And the determinants of antigenicity, the susceptibility of the tissue enzymes. The antigens must be susceptible to the tissue enzymes for the phenomena of antigen processing and antigen presentation. So antigens of virus and bacteria, and the surface of a bacteria, there will be an antigen, and this will be the determinant site of our immune system. Against these epitope or antigens, the antibody will be produced, and the produced antibody, it will be react on this particular region, and then it causes neutralization. Or based on the type of epitope, there will be different types of antigen antibody reaction. And the different epitopes on the bacteria reacting to the specific antibody. So one epitope, okay, so it never combines with all the antibody which is produced. And this we discussed during ELISA itself, antigen antibody are highly specific in nature. And antigen which will be containing one epitope. To this epitope, antibody it is produced, and that epitope, that antibody, it never goes and binds in the other epitopes of an antigen, or it may be microbes or bacteria. So if you have more than one epitope, each epitope will be produced one antibody, and each antibody will be producing against that particular epitope. So this is what the antibody, where it binds exactly. So this is the site why it makes this the amino acid a flexible region. So this is the region it is called as an antigen binding site. So if even it is a sequential antigen, or it may be a discontinuous non-sequential antigen also, by making conformational difference, the antibody definitely goes and binds into the antigen in the different peptide chain. And this is the region where we are making into the detecting antibody in ELISA, what we call it as a secondary antibody, where we are able to makes the antibody to bind with antigen antibody complex. And this is the region where in both arms, the antigen it goes and binds and it causes the neutralization. So this is a typical antibody structure where we have two chains. Okay, one it is called as a constant domain, and the other one it is called as the variable domain. Here H it stands for heavy chain, and the L it stands for the light chains. So constant domain at heavy region, and variable region at the heavy. So it will be, this is the antigen binding site. This flexibility it makes, or this the produced type, that is why the antigen it will be specifically bind with the one particular antibody. If there is any flexibility is there, and it will bind with the different epitopes what it is present in the antigen. So that flexibility it won't be there, and that is why antigen and antibody are highly specific in nature. And FAB, it is a fragment of antibody binding site. So that is one more. And FC, it is the fragment of crystallization. So antibody structure, it is again, it's another important 5-mark question, as like antigen. So antigen, so far it is not asked in the university examination, but antibody many times, what do you mean by antibody and types of antigen and antibody reaction? That is one more repeated 5-mark question in the university examination. So now I am going to explain about what do you mean by antibody. So antibodies are also called as an immunoglobulin or immunoglobulins, IgEs. Okay. So with the molecular weight which is having around 150 to 900 Dalton, it's constituted of majorly gamma globulin as a part of the blood protein. So they are soluble protein secreted by the plasma offsprings. And during B cell maturation, we discussed what is the role of plasma cell and primed B cell. And this produced antibody, wherever you have a soluble antigen, and this B cells, it the produced antibody, it goes and binds into that particular antigen and it causes the reaction. So this antibody, it inactivates the antigen by various antigen antibody reaction. The one reaction it is called complement fixation. So what is this complement fixation? In sense, the proteins it attaches to the antigen surface and cause the holes to form a cell lysis. The second reaction is called neutralization, where it binds to the specific site to prevent the attachment of this antigen into the target site. And the third one is agglutination, otherwise it is called as clumping. And this is what the best example, it is called the one which we discussed as a typhoid reduction, or even it goes for the blood grouping matching, where it is called as agglutination, when antigen it is containing more than one epitope, and that it causes the agglutination type of reaction. The agglutination and precipitation is the one, and forcing the insolubility and then settling out into the solution. Even precipitation reaction, if the antigen it is containing more than one epitope, okay, so that time you can see a precipitation reaction. So these are all the types of antibody and its molecular weight. I would have studied in detail in your biochemistry or even. So what are the types of antibody? Immunoglobulin M, otherwise it is called mu, and immunoglobulin G, it is called gamma, alpha, and delta, and eta. So these are all the antibody based on the type of immune response. So different antibodies it will be produced. And one interesting factor with the immunoglobulin A, assume an antigen which is not detected by our own immune system. This immunoglobulin A, it comes into an action, and what it causes, it coats an antigen, and that process is called as opsonization. Wherever there is a immunoglobulin A, our immune system, it goes and recognizes immediately that particular antigen, and later it will be treated effectively through our immune system. So that is the way the immunoglobulin A, it functions. It is containing two subclasses, immunoglobulin A1 and immunoglobulin A2. And immunoglobulin D and E, and during allergic reaction or toxic reaction, these two it secretes. So majorly immunoglobulin M and immunoglobulin gamma, it takes the complete responsibility of antigen and antibody reactions or neutralization or whatever the reactions which we discussed. So this, it may be an YOC question, what is the immunoglobulin? It involves in the opsonization reaction. And what do you mean by opsonization? When antigen it enters into our own body, if the immune system, our own immune system, and it is not recognized that antigen, this immunoglobulin A, it goes and coats that particular antigen, and it makes the rough coating. And after that, our immune system, it easily recognizes that particular antigen. Later it will be produced enough amount of antibody against that antigen. That process is called as opsonization. So the overall structure of immunoglobulin molecule is determined by either primary, secondary, tertiary, and quaternary organization of amino acid molecule. And the primary structure is just a sequence of amino acid that comprises the variable and constant region of heavy and light chain, as like in the previous slide I was showing the structure. And the secondary structure is formed by folding the polypeptide chain into series of beta pleated sheets. And the secondary structure is then folded into tertiary structure of compact globular domain. Finally, these globular domains of adjacent heavy and light chains interacts in the quaternary structure, forming a functional domains that enables binding site for antigen, and at the same time, it performs a number of biological functions. Two domains are found in light chain. One is a variable region and the other one is the constant region. In heavy chain also, one variable and constant region will be there. In one domain is found to be a variable domain and the other one it is in immunoglobulin A and immunoglobulin G and immunoglobulin D, three domains are found in the constant region CH1, CH2, and CH3, whereas immunoglobulin E and immunoglobulin M found domains are found in constant region. Okay. High region of H chain CH1, constant region H1, and constant region H2, and constant heavy chain 3 and constant heavy chain 4. There are four domains it is present in the immunoglobulin M. Fab region is the one region FAB I mentioned in the previous slide of when I show an antibody structure where this is the antigen binding is accomplished by an amino terminal. So you know an amino acids N terminal and C terminal, and the region and effector function of the carboxyl terminal of an antibody. So in an antibody molecule, two FAB regions are found and that binds to the antigen. FC region, it is called fragment of crystallization. FC region of immunoglobulin, it allows for interaction of the immune complex with other phagocytic cells as well as in the complement. So it takes part in the various biological functions that are determined by the amino acid sequences of each domains of the constant region, and many different forms of the fragment of crystallization receptor which is exists in the antibody. And the hinge region, this is a mu and delta and alpha heavy chain contains an extended peptide sequence between constant heavy chain and constant heavy chain 2, domain that has no homology with the other domain. This region is known as the hinge region. Hinge region is rich in proline residue and is flexible. Therefore, immunoglobulin G, D, A are flexible in nature. So this hinge region only we are changing into glycine-rich region, and so that we are preparing a secondary antibody that detects the antigen antibody complex. And these are all the types of antibody, how it will be available as a monomer or pentamer. Immunoglobulin G and D, it will be available as a monomer. Immunoglobulin E, this is the structure of immunoglobulin E. And where you have the binding site, and immunoglobulin A, it will be present and it will be available as a dimer. Okay, so the immunoglobulin A always it will be available as a dimer, and immunoglobulin M, it will be available as a pentamer. So when the antibody itself it is available, in what way it is available? Okay, so that the treatment of most of the antigens through immunoglobulin M, when it binds, it will be strongly reacted with the antigen. So that is why the first infection when it occurs, the first antibody if you see immunoglobulin G will be first produced, and followed by most of the common treatment. If you see the immunoglobulin M, it takes the responsibility for neutralizing the antigen. So this region it will be heavily treating with the antigen, so that the reaction or neutralization it will happen very easily. And now individually immunoglobulin G. So molecular weight is around 1,50,000 Dalton. It is containing heavy chain, and the gamma 53,000 Dalton. Immunoglobulin G is the most abundant class of immunoglobulin in the serum and constitute about 80% of the total serum of immunoglobulin. And immunoglobulin G molecule consists of 2 gamma heavy chain and 2 kappa and 2 lambda light chains. There are 4 subclasses of immunoglobulin G: immunoglobulin G1, G2, G3, and G4. It has the longest half-life among the other antibody. The half-life is around 23 days. And immunoglobulin G is the only, as like RBC and other cells, even antibodies are also having half-life. And immunoglobulin G is the only antibody that can cross placenta, and provide immunity even for the fetus up to 6 months of age. The immunity is known as a natural passive immunity. It can also activate the complement system. This is what the immunoglobulin G molecule. And each antibody we explain in detail with one diagram. And the biological function of immunoglobulin G is the major antibody produced in the secondary immune response. And immunoglobulin G, immunoglobulin G3, G4, it readily crosses the placenta, and it plays an important role in protecting the fetus. Immunoglobulin G3 is the most effective complement activator, followed by immunoglobulin G1, G2, and G4. and it is not able to activate the complement at all. And immunoglobulin G1 and G3 binds with a high affinity to fragment of crystallization receptor on phagocytic cell, and thus it mediates the opsonization. So that is what I explained to you in the previous slide of the immunoglobulin A. And immunoglobulin G helps in the bacterial immobilization from the location to another location. And immunoglobulin G, it neutralizes the toxin of virus. And immunoglobulin M, it is a molecular weight of 9 lakh Dalton, heavy chain type it is a mu, and 65,000 Dalton. So immunoglobulin M is secreted by the plasma cell and it exists in the pentameric form. And that's why it's a pentamer, and it is having 5 immunoglobulin monomer are linked together by a disulfide bond. It is called as the J sign. And there are 10 antigen binding site in the FAB in pentameric form. You know, each antibody is having 2 arms. So when you have a pentamer, 5 into 2, so there will be 10 antigen binding site in the immunoglobulin M molecule, but it cannot bind to 10 complete antigens due to the steric hindrance. It is the major antibody produced during the primary immune response, and monomeric form of immunoglobulin M is around 180,000 and is also expressed as membrane-bound receptor on the B cell. And immunoglobulin M is the dominant antibody which is produced in the primary immune response, while immunoglobulin G dominates in the secondary immune response. And immunoglobulin M is physically much larger than the other immunoglobulin. Mostly the immunoglobulin G and M is the major responsibility during our infection. And immunoglobulin A, it is the one that is having molecular weight of 320,000 Dalton, heavy chain type. It is alpha. It is containing 55,000 Dalton alpha chain. And immunoglobulin A, it constitutes around 10 to 15% in the total serum of immunoglobulin. And it is the predominant immunoglobulin in external secretions such as breast milk, saliva, tears, and the mucus of bronchial and genitourinary tract, as well as in the digestive tracts. Immunoglobulin A primarily existing as a monomeric form, but a dimeric, trimeric also it is available in some forms. It is immunoglobulin A, it is present. Immunoglobulin A in the blood, it occurs as a monomeric form, whereas those in the body secretion, it occurs in the form of dimeric or multimeric forms. So that is what I show you in the image. Always what you studied in the, what you seen in the first slide, it is just normal immunoglobulin structure, but how it exists, each immunoglobulin, it exists in the different forms from monomer to pentamer. And the dimeric form of immunoglobulin A contains the J chain and the secretory chain. And secretory chain helps in the transcytosis. And immunoglobulin A, it can cross the epithelial layer and enter into the body secretion. Because of that monomeric unit, no, it can easily crosses the placenta also. And this process of crossing the epithelial layer by immunoglobulin A, it is known as the transcytosis. And there are 2 subclasses in the immunoglobulin A, immunoglobulin A1 and immunoglobulin A2. Biological function, immunoglobulin A can cross the epithelial layer and it enters into the body secretion and provides local immunity in the gastrointestinal tract as well as respiratory tract and genital tract. In the body secretion, immunoglobulin A, it neutralizes the viruses and prevent the attachment on the host surface. Immunoglobulin D, molecular weight of 180,000 Dalton and heavy chain type, it is delta with the 70,000 Dalton. Immunoglobulin D is present extremely in the low concentration and it constitutes only 0.2% of the total serum of immunoglobulin, and immunoglobulin D together with immunoglobulin M is the major membrane-bound immunoglobulin expressed on the mature B cells. There are 2 subclasses, immunoglobulin D1 and D2. Immunoglobulin D plays important role in the maturation and proliferation of the B cells more than functioning with the antigen. And immunoglobulin E, it's a molecular weight of 2 lakhs Dalton and heavy chain type is epsilon, having 73,000 Dalton molecular weight. Immunoglobulin E, it accounts for 0.3% only in total serum immunoglobulin. Immunoglobulin is also known as the reagenic antibody due to its involvement of the allergic reaction. So whenever there is an allergic reaction, the first antibody what it secretes, it's called as the immunoglobulin E. So this immunoglobulin E, it mediates the immediate hypersensitivity reactions, and it is responsible for the symptoms like hay fever, asthma, anaphylactic shock. So hypersensitivity reaction, I'll going to explain in my next presentation. By the time you can understand how important this immunoglobulin E understanding. And the level of immunoglobulin E antibody in the blood of normal individual is very low, and its level is increases during parasitic infection and also during allergic reactions. So that's all about the antigen as well as antibody. There is one more important uh, composition or a repeated 5 mark question. What are all the various types of antigen and antibody reactions? So already, as I told you, there are a few types already we discussed in the introduction slide of an antibody. And in this case, you can elaborate, you can mention it like complement fixation, precipitation, agglutination. And ELISA is also one of the antigen and antibody reaction. So if you are not thorough with complement fixation, precipitation, or agglutination, you can start elaborating ELISA. Even ELISA is elaborating about the direct, indirect, and sandwich and the competitive ELISA. That is also considered equally for a 5 mark question. So very well, you can highlight about ELISA for antigen antibody reactions. So what is this complement of fixation or precipitation reaction? Antibody and soluble antigens, it interact in the aqueous solution and it forms a lattice that eventually develops into a visible precipitate. For this precipitation reaction only, you have to have that multivalent or polyvalent epitopes should be present on an antigen. Antibody that aggregate the soluble antigens are called as the precipitants. Although formation of soluble antigen antibody complex occurs within a minutes, the formation of the visible precipitate occurs more slowly and often it takes sometimes it may be day or 1 day or two to reach the complete precipitation reaction. Formation of antigen antibody lattice it depends on the valency of both the antibody and antigen reaction. So that's what the number of epitopes what it is present on the antigen site. And the antibody must be bivalent. A precipitate will not form with a monovalent fragment of antibody binding site fragments in antibody. The antigen must be either bivalent or polyvalent, and that it is must to have at least 2 copies of the same epitope or have a different epitopes that react with a different antibody present in the polyclonal antiserum. So that is what we discussed in detail about in antigen, what is the number of epitope and what it causes. Same number of epitope or different epitope with a different antibody. Only then the precipitation reaction we can see. And precipitation reaction in fluids yields a precipitation curve based on the amount of antibody which occurs in a different time interval. So when you have antibodies which is produced in the layer during infection and based on the availability of the antibody zones, and this is divided this precipitation zone and the antibody excess zone and equivalence zone and antibody antigen excess zone. So this is what the precipitation curve when you draw against the antigen and antibody reaction, you will be getting a curve like this. And based on this curve, and you can understand where antigen rich region or antibody, how it binds or how it causes the precipitation reaction. And precipitation reaction in gels, it yield the visible precipitating line. And the immune precipitates can form not only in the solution, but also it is in the agar matrix. And 2 types of immunodiffusion it occurs. It can be used to determine a relative concentration of the antibody or antigens. To compare antigens are determined the relative purity of the antigen preparation during our ELISA or some commercialized or standardized preparation. They are using this precipitation reaction as a standard or quality control of either antigen or antibody. So they are radial immunodiffusion assay and double immunodiffusion assay. Otherwise, it is called as the Aptoloni method. Both are carried out in the semi-solid medium, such as an agar. Agar containing medium, you can perform this test. And this is what the antigen, maybe what we did in the disk diffusion assay of antibiotic, the same you can perform it with the help of antigen and antibody. So when the antibody is introduced, when antigen is introduced, how fast the precipitation reaction that is occurred in the cell of diffusion is called as the aptolonic assay. Through this, you can find out the quality of the antigen or the quality of the antibody. So in radial immunodiffusion, an antigen sample is placed in a well and allowed to diffuse into the agar containing suitable dilution of an antiserum, which is containing an antibody. Same way of the microbial and antibiotics. So here we are using antigen and antibody. So same way, same methodology like a disk diffusion. As the antigen diffuses into the agar, the region of equivalence is established and a ring of precipitation line. You can see a precipitation ring and forms in around the well. Okay, the area of the precipitation ring is proportional to the concentration of antigen. The antigen is less and the precipitation reaction, that ring, it will be very less, where based on the antibody concentration, the ring, it will be vary. So by comparing the area of the precipitation ring with a standard curve and the precipitation area of the known concentration, the concentration of the antigen, what is present in the commercially produced sample, that very well we can determine. So we have this is a double immunodiffusion assay where both antigen and antibody are added, in which point where there is a precipitation reaction occurs, and that is the point you can take it as a precipitin reaction. So where the distance is already, we have a standard value, from that you can easily find out what is the precipitation reaction, and in what way you can standardize your quality